BrainTACO, an explorable multi-scale multimodal brain transcriptomic and connectivity data resource. Florian Ganglberger, Dominic Kargl, Markus Töpfer, Julien Hernandez-Lallement, Nathan Lawless, Francesc Fernandez-Albert, Wulf Haubensak, and Katja Bühler. Communications Biology 7, article number 730, 2024. Cite this article. 3,485 accesses. 1 Altmetric. Metrics. Abstract. Exploring the relationships between genes and brain circuitry can be accelerated by joint analysis of heterogeneous datasets from 3D imaging data, anatomical data, as well as brain networks at varying scales, resolutions, and modalities. Generating an integrated view beyond the individual resource's original purpose requires the fusion of these data to a common space and a visualization that bridges the gap across scales. However, despite ever-expanding datasets, few platforms for integration and exploration of this heterogeneous data exist. To this end, we present the BrainTACO, Brain Transcriptomic and Connectivity Data resource, a selection of heterogeneous and multi-scale neurobiological data spatially mapped onto a common, hierarchical reference space, combined via a holistic data integration scheme. To access BrainTACO, we extended BrainTrawler, a web-based visual analytics framework for spatial neurobiological data, with comparative visualizations of multiple resources. This enables gene expression dissection of brain networks with, to the best of our knowledge, an unprecedented coverage and allows for the identification of potential genetic drivers of connectivity in both mice and humans that may contribute to the discovery of disconnectivity phenotypes. Hence, BrainTACO reduces the need for time-consuming manual data aggregation often required for computational analyses in script-based toolboxes and supports neuroscientists by directly leveraging the data instead of preparing it. Similar content being viewed by others. Neuromaps, structural and functional interpretation of brain maps. Article Open Access, October 6, 2022. Open datasets and code for multi-scale relations on structure, function, and neurogenetics in the human brain. Article Open Access, February 29, 2024. SODB facilitates comprehensive exploration of spatial omics data. Article February 16, 2023. Introduction. An increasing amount of evidence suggests that human behaviors and their impairments in psychiatric disorders are better understood via a multimodal data integration approach than by analyzing individual neurobiological measures 1, 2. Many insights into the brain's functional organization and neuronal mechanisms were sparked by collecting and interpreting spatially organized histology, cellular composition, connectivity, and activity data. For instance, the entry points for modern neuroscientific experimental workflows are brain regions, i.e. part of a specific neuronal circuit thought to be involved in a brain function or behavior, whose gene expressions and functional connectivity patterns are studied to understand the circuit dynamics underlying a behavior. That information can then be used to identify targets in the brain that could be modulated by psychoactive drugs in cases of psychiatric symptomatology 3. Thus, integrating both functional connectivity and omics data modalities is instrumental to better understanding the biological underpinnings of behaviors and their deficits. 4. Recent advances in neuroimaging allowed big brain initiatives and consortia to create vast resources 5, 6, 7 of data, which could be mined for additional and deeper insights. However, collecting these data from different sources for comparison and exploration leads to several challenges, as they are acquired in different systems and can vary in resolution, anatomical scale, or sampling density. A mandatory first step is to map the data onto a common reference space to ensure alignment for imaging data and annotation using the same brain region ontology 8. In an alternative approach, such as mapping the data to the smallest common denominator, e.g. major anatomical brain regions 9, one loses granularity and specificity, rendering the data potentially less representative. Neuroscience studies that use a combination of omics, imaging, anatomical, and connectivity data often require extensive analytical workflows, including mapping to a common reference space 8, manual data aggregation 9, and statistical analysis. This typically requires the expertise of a bioinformatician to find patterns that might relate to a given behavior 9, 10, 11, 12, 13, 14, 15, 16. The term big refers to the amount, vast image collections, many datasets, and/or size, high-resolution imaging/network data of the data, which is too extensive to be analyzed with traditional methods. Here, visual analytics tools bridge this gap by enabling neuroscientists to interactively browse vast data collections, visualize complex relationships, and link different types of data. Many neuroscientific resources for transcriptomic data provide interactive, web-based visualizations for access and exploration. A comprehensive collection of such websites has been provided by Kargl et al. 17. While providing access to scientists without the need for advanced computational expertise, they are primarily suited for single datasets, i.e., they rarely provide workflows across multiple datasets and modalities. One notable exception is the Cyber Explorer via EBRA-INS 18, which combines structural connectivity, fiber tracks, 19 with microarray-based gene expression data 20, 21. Another relevant tool, although not web-based, is Brain Explorer 22, 23, which enables the retrieval of structural connectivity from the Allen Mouse Brain Connectivity Atlas 24 in combination with in situ hybridization data 25. Nevertheless, in their current state both, Cyber Explorer and Brain Explorer are limited to one connectivity and one transcriptomic dataset each, while lacking support for next-generation sequencing data. In this paper, we present a holistic data integration scheme to map heterogeneous brain data across scales, spatial and anatomical resolutions, as well as sampling and acquisition types, Figure 1. BrainTACO, Brain Transcriptomic and Connectivity Data, is a resource that includes bulk and single-cell/nucleus RNA sequencing, in situ hybridization, and microarray-based transcriptomics data, as well as structural and functional connectivity mapped onto common hierarchical reference spaces. To make BrainTACO accessible, we built onto previous work, BrainTrawler 26, a tool for visualizing volumetric, geometry, and connectivity data simultaneously in 3D rendering and 2D slice views, which can iteratively integrate additional heterogeneous datasets from the community and across species. We extended BrainTrawler to integrate, store, and query datasets from various resources via our spatial indexing-based data structure 27. It enables automatic aggregation and interactive exploration of large-scale, high-resolution spatial connectivity. 7, 24, and image collections of gene expression data 25 on different scales. We extended our data structure to integrate sample-based region-level datasets, i.e. sampled from brain regions, such as microarray gene expression data or count matrices from RNA sequencing. Here, it is possible to aggregate samples on individual dataset level by user-defined regions of interest in real time so that different datasets can be compared on the same anatomical level, independent of their original resolution and scale. Figure 1. Mapping data of different resolution and scales to a common reference space. 1. Voxel-level data is mapped to the voxel-level reference space by image registration. 2. Region-level data, e.g. RNA sequencing data, is mapped via a hierarchical brain region ontology, i.e. standardized hierarchical brain parcellation with region annotation, with voxel-level parcellation to the reference space. 3. All data mapped to the reference space can be either retrieved on the resolution of the reference space, data is up, or downsampled via nearest neighbor interpolation, or on every other region level of the hierarchy. Full-size image. Via a web interface, BrainTACO can be used to dissect brain connectivity interactively with a wealth of transcriptomic data, similar to Ganglberger et al. S26 previous approach for in situ hybridization data only. To account for the increased number of datasets, as well as the increased complexity of the datasets itself, e.g. samples from multiple cell types, developmental states, etc., we added additional comparative exploration functions. Here, we facilitated visualization techniques such as heat maps, small multiples 28, and parallel coordinates to identify gene expression patterns across datasets and categorical information, cell types, phenotypes, developmental stage, interactivity on arbitrary levels of anatomical detail. This enables neuroscientists a view on the data, tailored to their research focus, and without the need for programming knowledge. Our resource closes the gap in current interactive analytical tools by combining gene expression, structural, and functional relationships at the microscopic, mesoscopic, and macroscopic level. This is achieved by the following: a hierarchical brain ontology-based integration scheme, i.e. brain parcellation with standardized hierarchical region annotation to access neurobiological, spatially mapped data across resolution, anatomical scale, or sampling density. A collection of publicly available gene expression, in situ hybridization, microarray, bulk and single-cell/nucleus RNA sequencing, and connectivity, structural and functional resting state. Datasets covering major anatomical brain regions mapped onto common hierarchical reference spaces. The data's original annotation is stored and made transparent, data provenance. An intuitive web interface for comparative visualization to access the BrainTACO resource in real time without programming knowledge. Results: integrating multimodal, multi-scale resources. To create a resource, resource of brain-wide gene expression (21), both already mapped to the reference spaces. Figure 2. Dataset coverage over major anatomical brain regions. Full-size image. RNA sequencing datasets were leveraged using TPM, transcripts per million. Second, interdataset comparability was assessed on rank level, i.e., whether the general order of genes by their TPM/CPM was consistent across datasets. Therefore, we visualized heat maps that show Spearman rank-based correlation over all genes of mouse and human datasets that cover matching brain regions and cell types, e.g. Bhattacharjee et al. 34 and Yao et al. 36, as seen in Figure 3. Black boxes in the heat maps mark correlations of matching cell types in different datasets, indicating consistent ranking of gene expression. Hence, comparability of relative gene expression between datasets can be assumed. While the comparison of co-expressed genes via cell type and region specificity would provide further mechanistic insights about gene regulation, this is in general limited because of the absence of matching references. i.e. region specificity would require datasets covering the same brain regions, cell type specificity the same cell types. Types. Figure 3. He-maps showing the Spearman rank-based correlation over all genes of mouse and human datasets that cover the same brain regions and cell types. Black boxes mark correlations of the same cell types in different datasets. On average, their correlation is higher than for not matching cell types, which indicates that the ranking of gene expression is consistent across datasets, one-sided Wilcoxon test, all p-values less than or equal to 0.05, except mouse visual areas. Neuronal subtypes in mouse visual areas were already so similar within datasets, all correlation greater than or equal to 0.95, that there was no significant difference across cell types. Full-size image. For details about the mapping of the datasets, as well as the pre-processing and normalization, see the methods, section data mapping and querying, and data pre-processing and normalization. Supplementary data 2 and supplementary data 4 provide additional information and code. Mapping to a common reference space. The joint exploration of spatial datasets from different resources requires the data to be aligned to a common space 8. This space acts as a reference so that spatial locations, such as coordinates or brain region annotations, have the same meaning across datasets. In neuroscience, commonly used reference spaces are typically defined by an anatomical reference template 2930, a set of structural images that have been combined, e.g. via image registration, to a complete representation of the brain for a group of specimen or a species. Imaging data, i.e., data that is divided by a 2D/3D grid into pixels/voxels that represent measurements at their respective positions, can then be aligned onto a template by image registration. This involves image transformation and warping to establish voxel-level correspondence. Figure 1. As templates, we use the Allen Mouse Brain Coordinate Framework 29, 0.1 mm resolution, and the ICBM 152 MNI Space Template 30, 1 mm resolution, based on their widespread use and availability 55. In principle, there is no limitation to specific templates. For spatial data that is not derived from imaging, i.e. measurements that have only been generated for specific brain regions, a different approach is needed. Here, we utilized hierarchical ontologies of brain regions, a formal representation of knowledge about species-specific brain anatomy 56, Figure 1, i.e., which brain regions it is composed of and how these brain regions are subdivided. Hierarchically. The Allen Institute provides ontologies for both mouse and human 29, 57, 58 that include a mapping onto our respective reference spaces, i.e., a direct correspondence between each coordinate of the reference space to a brain region in the ontology. Since datasets are not necessarily annotated with the same ontology and on the same hierarchical level, they cannot be compared across anatomical scales and resolution directly. Hence, we matched brain region annotations to the corresponding brain regions in the ontology. An outline of this process is shown in Figure 1. Details can be found in the methods, section data, mapping, and querying. Note that these mappings are made explicit in our resource's user interface to ensure transparency and, as a consequence, allow for quality control. The distributed nature of brain functions across brain networks and gene sets necessitated a thorough exploration of spatial gene expression in the context of brain structural and/or functional connectivity. To this end, we integrated high-resolution imaging data of structural connectivity for mouse, 24, and resting state functional connectivity for human, 7. Structural connectivity, which describes how brain areas are physically connected via axonal projections, was originally imaged at a 100 micrometers resolution 24. The human resting state functional connectivity, on the other hand, describes brain regions that are linked by correlated activity. This data, sourced from the WUMIN Human Connectome Project 7, represents the group average dense, voxel-level correlation of the resting state bold signal of 820 subjects. We selected both the mouse structural connectivity data and the human resting state functional connectivity data for their high resolution and compatibility with the common reference spaces. See section data, pre-processing, and normalization for more details. Interactive access and exploration. BrainTrawler was one of the first iterations of an interactive, web-based visual analytics framework 26. Originally, it was designed to explore large-scale brain connectivity data, such as structural connectivity 24, and to dissect these connections on gene expression level in the mouse brain. This was achieved by providing a visual analytics workflow to identify which genes are expressed in either the source or target regions of these connections, based on spatially mapped gene expression data of 20.000 genes in the Allen mouse brain, Atlas 25. Interactivity was achieved by facilitating spatial indexing on volumetric images 59 for the spatially mapped gene expression data, as well as developing a data structure for real-time aggregation of connectivity data with billions of connections 27. We expand on this effort to handle large-scale transcriptomic datasets for mouse and human to not only show where genes are expressed, but also how expression differs between cell types and developmental or physiological conditions. We therefore build a spatial database of RNA sequencing and microarray-based gene expression datasets, including the datasets described in the previous section. This spatial database utilizes spatial indexing for aggregating gene expression of datasets in real time that were aligned to brain regions/voxels of the reference space. To this end, datasets including their metadata, e.g. cell type annotations, age, phenotype, etc., were sorted based on their spatial location in the brain. See methods, section spatial indexing for details. The exploration of gene expression related to brain connections works in an analogous manner as previously presented in Ganglberger et al. 26. See Figure 4. First, the user defines a volume of interest, VOI, which can be either an arbitrary manually defined area via a brush drawing tool or a brain region. Figure 4. A. Yellow area. For this VOI, a gene expression query can be performed, which computes the mean expression of all datasets that have been aligned to the reference space within the VOI. Results can be refined via a user-defined filter, i.e., selected meta-properties data such as certain cell types, phenotypes, and others, details in the methods, section data mapping, and querying. This approach offers a simplified representation of complex gene expression patterns within specific cell types and brain regions, enabling an accessible visual analytics approach for comparing key differences without losing essential information. To provide differential gene expression insights in relation to other cell types or the whole brain, cell type specificity, i.e. gene expression of a cell type versus other cell types, and region specificity, i.e. gene expression of a VOI versus the whole brain, queries can also be applied, offering a more comprehensive understanding of the gene expression landscape. See section data mapping and querying for details. Figure 4. Exemplary gene expression dissection of a structural connection, showcasing the process of defining volumes of interest, VOI, analyzing structural connections, and comparing gene expression patterns. Outgoing structural connections, red, from a user-selected part of the thalamus, yellow, representing the source VOI of the connection. This VOI can be an arbitrary manually defined area or a specific brain region. Be target VOI, yellow, of the structural connections, red, representing the strongest targets of the aggregated connections. See gene expression comparison in a parallel coordinates system for source VOI, red axis, 1 for astrocytes in the Zyzel_201837 dataset, 1 for gene expression in the line _2007 dataset, and target VOI, green axis, gene expression in the line _2007 dataset. Astrocytes in the Badature G_201934 and Yao _2020 datasets 36. Using different datasets and cell types. Each horizontal line represents a gene, with its position on the axis indicating the level of gene expression. The axes represent the results of gene expression queries, allowing for comparison and filtering of multiple gene lists. An example subset of genes with low astrocytes expression in the source VOI and high astrocytes expression in the target VOI is selected, demonstrating the potential for gene expression dissection of structural connections. Full-size image. The result of such a queries are lists of genes with the aggregated gene expression. Figure 4c shows how multiple queries results can then be compared in a parallel coordinate system, which allows filtering multiple gene lists by their gene expression. Each axis in the figure represents the result of a gene expression query and, as a consequence, the level of gene expression in the query regions. Each plot line represents a gene. A selection/filtering of genes, shown in the table in the lower part of figure 4c, with specific gene expression patterns can be made drawing brushes on an axis. Since queries of different VIs can be compared, one can use this on the source and target areas from connectivity data for gene expression dissection. Figure 4a shows the aggregated outgoing structural connectivity of the VOI in red. While the yellow VOI in figure 4a represents the source, the yellow area in figure 4. B represents the strongest targets of the aggregated connections. A comparison of the gene expression of source and target VOI can be seen in Fig. For C, here, the axes labeled in red are results of gene expression queries at the source VOI, green ones at the target VOI, performed for different exemplarily selected datasets and cell types. The increase in available datasets and their inherent complexity, e.g. samples from multiple cell types, developmental states, etc., makes it necessary to perform large amount of expression queries to cover all available information for genes of interest. Hence, we extended BrainTrawler's capability to visualize gene expression of multiple resources jointly by developing a lightweight interface, BrainTrawler Lite. BrainTrawler Lite's basic user interface element is a heat map of the dataset coverage, figure 5a. Each heat map tile represents the sample size/image quantity distribution of a certain dataset, rows, for a certain brain region, columns, similar to figure 2. By clicking on a heat map tile, these data can be selected for further investigation, either on a gene set level, by entering a list of genes, figure 5b, or on a genome-wide level, figure 5. C. Analogously to a gene expression query. Results can be exported as images or as tabulated text files for later use or for sharing. For more details, see Methods, section BrainTrawler Lite. Figure 5. BrainTrawler Lite interface for comparative visualization of gene expression across datasets. The dataset coverage heat map shows the distribution of sample size/image quantity across brain regions, columns, and datasets, rows, subdivided by metadata attributes such as cell types, phenotypes, etc. Brain regions and metadata categories can be adapted via tree-like UI elements on the sides. The tooltip shows the exact composition, sample/image count, metadata categories, etc., of the respective heat map tile. Orange tiles shows a selection of data for gene expression visualization in B, C. B gene expression heat maps of 5 selected genes. Rows and columns represent the selected orange tiles in the dataset coverage heat map in A. Color scales are separated per dataset between 0 and the maximum value shown for all selected genes. The right side shows the expression of genes for each dataset separately as small multiples. The left side shows one selected gene with more details, labels, values, etc. Gray tiles are missing data. C. Parallel coordinate system showing the gene expression of all genes in the selected dataset on axes, each representing the average expression of genes, blue lines, of the samples/images of each selected orange tile in the dataset coverage. In A, via drawing brushes on the axes, the genes in the parallel coordinates system can be filtered. Filtered genes are shown below in a table. Full-size image. Relating gene expression and connectivity across species uncovers genes and mechanisms for human functional connectivity. The functional FC and structural connectivity SC of the brain are viewed as a major determinant of cognitive function across species. Altered connection topology and intensity of brain areas commonly correlate to psychiatric conditions such as the autism spectrum and schizophrenia, suggesting that disconnectivity might lie at the core of these conditions 60, 61, 62. Alongside, genome-wide association studies have discovered genetic loci and polymorphisms associated with these psychiatric conditions, indicating that the relationship between the connectivity of a given brain area and its gene expression likely harbors valuable information on wiring principles of the brain 63, 64. To bridge these domains, the combination of connectomic and gene expression data is a promising approach to discover genetic etiologies and emerging mechanisms that drive regional efferent and afferent connectivity underlying connectopathies, conditions associated with aberrant brain connection topology 65. The increasing abundance of cell-type specific, gene expression, and connectivity data in the mouse is a promising avenue to discover genetic susceptibilities and mechanisms affecting the connectome, with great translational potential. Thus, identifying genetic drivers of FC in humans is of great interest, as it may provide entry points into therapeutic interventions to ameliorate disease burden. In the context of psychiatry, the insular cortex is of special interest as a core hub in regulating large-scale brain networks in humans 66 and rodents 67. It is also involved in interoceptive, cognitive, and affective processes 68, 69. Notably, insular functional disconnectivity is an indicator of common psychiatric conditions 70, 71. We segmented the insula into its granular and granular portions, as they are anatomically and functionally distinct 72. While the posterior granular insula, GI, is a primary sensory area with rich afferents for interoceptive information, the anterior agranular insula, AI, represents an associative area with increasing multimodal integration 68. Therefore, this system is an ideal model area to discover novel genetic factors shaping the connectivity of cortical areas of distinct architecture, a granular versus granular insula, in a highly relevant translational setting. First, to allow for optimal cross-species inferences, we selected consensus areas between the rodent and human brain, covering 10 major subcortical regions, supplementary data 5. Next, source and target connectivity data with these areas were sampled for the AI, combined L_granular insular area, dorsal part, and L_granular insular area, ventral part, and GI, L_visceral area, supplementary figure 1, left. Because the Human Allen Brain Atlas, ABA, does not discern by granularity, human FC data for AI and GI with the consensus areas were sampled by brushing a granular and granular areas of the short and long insular gyri. Supplementary figure 1, right, according to 73. Within species analysis shows a correlation between source and target SC in the mouse GI, but not in the AI, figure 6a. Overall, AI and GI connectivity is correlated in rodents and humans, supplementary figure 1. Figure 6. Relating regional gene expression with connectivity across species. Relating gene expression and connectivity across species uncovers genes and mechanisms for human. The functional FC and structural connectivity SC of the brain are viewed as a major determinant of cognitive function across species. Altered connection topology and intensity of brain areas commonly correlate to psychiatric conditions such as the autism spectrum and schizophrenia, suggesting that disconnectivity might lie at the core of these conditions 60, 61, 62. Alongside, genome-wide association studies have discovered genetic loci and polymorphisms associated with these psychiatric conditions, indicating that the relationship between the connectivity of a given brain area and its gene expression likely harbors valuable information on wiring principles of the brain's 63, 64. To bridge these domains, the combination of connectomic and gene expression data is a promising approach to discover genetic etiologies and emerging mechanisms that drive regional efferent and afferent connectivity underlying connectopathies, conditions associated with aberrant brain connection topology 65. The increasing abundance of cell-type specific, gene expression and connectivity data in the mouse is a promising avenue to discover genetic susceptibilities and mechanisms affecting the connectome, with great translational potential. Thus, identifying genetic drivers of FC in humans is of great interest, as it may provide entry points into therapeutic interventions to ameliorate disease burden. In the context of psychiatry, the insular cortex is of special interest as a core hub in regulating large-scale brain networks in humans 66 and rodents 67. It is also involved in interoceptive, cognitive, and affective processes 68, 69. Notably, insular functional disconnectivity is an indicator of common psychiatric conditions 70, 71. We segmented the insula into its granular and granular portions, as they are anatomically and functionally distinct 72. While the posterior granular insula, GI, is a primary sensory area with rich afferents for interoceptive information, the anterior agranular insula, AI, represents an associative area with increasing multimodal integration 68. Therefore, this system is an ideal model area to discover novel genetic factors shaping the connectivity of cortical areas of distinct architecture, a granular versus granular insula, in a highly relevant translational setting. First, to allow for optimal cross-species inferences, we selected consensus areas between the rodent and human brain, covering 10 major subcortical regions, supplementary data 5. Next, source and target connectivity data with these areas were sampled for the AI, combined L_granular insular area, dorsal part, and L_granular insular area, ventral part, and GI, L_visceral area, supplementary figure 1, left. Because the Human Allen Brain Atlas, ABA, does not discern by granularity, human FC data for AI and GI with the consensus areas were sampled by brushing a granular and granular areas of the short and long insular gyri. Supplementary figure 1, right, according to 73. Within species analysis shows a correlation between source and target SC in the mouse GI, but not in the AI, figure 6a. Overall, AI and GI connectivity is correlated in rodents and humans, supplementary figure 1. Although to different extents between sources and targets. As expected, human FC is not significantly correlated to mouse SC, suggesting relevant functional differences between species and/or connection modality, Fig. 6b. Figure 6. Relating regional gene expression with connectivity across species identifies potential connectivity driver genes, where cell-type specific analysis reveals a conserved inhibitory mechanism across species and connection modality. A mouse source and target connectivity is significantly correlated for GI, Spearman, art equals 0.65, p-value equals 0.04, but not for AI. Art equals 0.08, p-value equals 0.82. B-mouse SC source and target connectivity are not significantly correlated to human FC, Spearman. AI. Source R equals 0.22, p-value equals 0.54. Target R equals 0.22, p-value equals 0.54. GI. Source R equals 0.22, p-value equals 0.54. Target R equals minus 0.05, p-value equals 0.89. See overlap of respective top and bottom 1% of genes correlated to connectivity across species, mouse SC source/target, and human FC. D. Brain-related associations of overlapping genes in C, total of 26 genes for AI, 21 genes for GI, C. Supplementary data 7 for summary. E. Cumulative distribution of correlation coefficients resulting from the correlation of cell type specific gene expression and mouse SC sources/targets across excitatory and inhibitory cell types for AI and GI. Kolmogorov, Smirnov tests revealed significant differences for AI and GI, P. Full-size image. It is established that brain oscillations are governed by recurrent inhibitory In summary, despite the fact that human FC and mouse SC are not discussion, we created a discovery framework that utilizes data from current popular large-scale genetic and brain network initiatives to rapidly screen for neural circuitry underlying specific brain functions, behaviors, or psychiatric symptoms at comparably low computing costs. Our platform has several implications for both basic and biomedical research and may directly impact subsequent circuit genetic experiments such as electrophysiology, opto-dash, and pharmacogenetics. When performed at large scale with behavior-specific genes, our approach has the potential to refine knowledge about the functional organization of the brain beyond simple anatomical domains. Importantly, our methodology for generating and exploiting data resources could be applied to other model organisms for which spatially mapped gene expression, network, and genetic information is, or will be, available. For example, fruit flies or zebrafish. Using our platform, we show Inevitably, BrainTACO has some limitations. First of all, in its current state, it does not cover the full brain on a single cell/nucleus level. To provide a resource as versatile as possible, we focused on covering the brain at least the level of most major anatomical brain regions, so that neuroscientists will likely find data related to their own research focus. This was not entirely possible for human datasets, e.g. amygdala, thalamus, hypothalamus, due to a lack of studies covering these areas. In principle, BrainTACO is not static and can be extended with new datasets. Future studies, as well as further improvements of technologies such as spatial transcriptomics, will help to close this gap. Another limitation is that it is in general not reasonable to compare absolute gene expression values, even normalized ones, e.g. TPM, CPM, across datasets 53, 54, especially with respect to batch effects. We circumvent this issue by providing dataset-specific color scales for gene expression he-maps and advise to compare absolute gene expression only relative to other genes within the same dataset. As an alternative, we provide gene ranks for gene expression queries, i.e., their relative position in a list of genes sorted by their expression. This relative measure, in terms of how is a gene's expression ranked relative to all other genes for a certain cell type/brain region, acts as a normalization to adjust for batch effects and other sources of technical variation, but may not completely eliminate them. Bulk RNA sequencing data. To fill gaps in subcortical single-cell/nucleus RNA sequencing data for the human, we integrated two bulk RNA sequencing datasets from the GTX and BrainSpan Consortia 4041. GTX data was downloaded from the GTX portal, https://gtxportal.or, in version 8 as gene TPM, transcripts per million. BrainSpan data was obtained from the BrainSpan portal, https://www.brainspan.org, as normalized RPKM, reads per kilobase of transcript, expression values, and converted to TPM according to Zhao et al. 53. We applied log2 normalization, using an offset of 1 to account for zeros, to both datasets, similar to single-cell/nucleus RNA sequencing data processing. Processing. Microarray gene expression data. Microarray gene expression data was retrieved from the Allen Human Brain Atlas by Horowitz et al. 2021 via the Allen Brain Atlas API. This data assembled gene expressions from 3702 samples of 6 donors, labeled with their according brain region in the ontology provided by the Allen Institute 57, which ensures equivalent scaling across donors. We normalized gene expression values based on an outlier-robust sigmoid function before rescaling the normalized values to a unit interval, 0 to 1, as suggested by Arnott-Kovicit et al. 82. In situ hybridization data. Whole brain gene expression in situ hybridization data was retrieved from the Allen Brain Atlas API as volumetric images for 19,479 genes. To create these volumetric images, the Allen Brain Atlas divided the in situ hybridization slice images on cellular resolution into a 200 micrometers resolution grid. For each grid division, expression energy was computed, i.e. the sum of the expression intensity of all pixels within the division, divided by the sum of pixels within the division 83. The expression energy for all grid divisions can then be seen as a 200 micrometers resolution volumetric image. To make this data available in BrainTrawler, we logged2, normalized the data, and encoded them as 8-bit volumes with a size of 155 kilobytes each, 3 GB in total. Structural connectivity data. Structural connectivity was generated similar to previous publications 26, 27, 84. Here, the connectivity was retrieved from the Allen Brain Atlas API as volumetric images, showing structural connectivity of 2,173 injection sites to their target sites 24. These 2173 images were generated on a 100 micrometers resolution by labeled RAV tracers via serial 2-photon tomography 24. For each image, the injection site is given by coordinates in the reference space defined by the Allen Mousebrain Coordinate Framework 29. And an injection volume, depicting the volume around the injection site affected by the tracer. Hence, the connectivity for an injection site is defined by all voxels within its injection volume. For every voxel in the reference space, we took the connectivity from the covering injection volume. If a voxel was covered by multiple injection volumes and therefore by multiple injection sites, we combined them by taking the maximum connectivity for each target. To compensate for low count of injection sites on the left hemisphere, we mirrored the connectivity, effectively doubling the original 2,173 injection sites to 4,346. To minimize the amount of false positive connections, the data was thresholded by values less than 10. Minus 4.5 according to O et al. 24, extended data figure 7. The result was a dense 67, 500 times 500,000 structural connectome, 67500 source voxels, covering injection volumes with 500,000 target voxels within the mouse brain, with 90 GB stored in a CSV format. Resting state functional connectivity data. Resting state functional connectivity data was downloaded from the WUMIN Human Connectome Project 7 via the Connectome DB, https://db.humanconnectome.org. We specifically chose the human resting state functional connectivity data over the structural connectivity diffusion tensor imaging data provided by the Human Connectome Project 7, because it was more readily pre-processed and provided higher resolution than region, level 85, better showcasing the capability of BrainTrawler to support even voxel-level connectivity. The data was available as an average functional connectivity matrix of 820 subjects, given as a dense 90,000 times 90,000 functional connectome in gray ordinate space 7, where a gray ordinate is either a voxel, subcortical gray matter, or a surface vertex, cerebral cortex. The Human Connectome Project provides the cortical surface, vertices, of the 152 MNI template 30 in gray ordinate space. We use the Connectome Workbench Platform 7. To extract the surface vertices coordinates in MNI space, see script get voxelan vertices to MNI space mapping, M provided in supplementary data. 1. To finally map the functional connectivity matrix to our 1 mm resolution ICBM 152 MNI space template reference space, We assigned every voxel of the template with the closest cortical surface vertex coordinate or subcortical voxel coordinate. This resulted in a dense 87,000 times 87,000 functional connectome with 45 GB stored as CSV. Data mapping and querying. Imaging data shown in this paper, namely in situ hybridization data 25. Axonal projection connectivity 24 and resting state functional connectivity 7 was already aligned to the reference spaces used for this resource 29, 57, 58. Novel datasets can be aligned via tools such as the Quint Workflow 86 or the ANTS Framework 87. Data that does not meet the resolution of the reference space can be up or downsampled via nearest neighbor interpolation. This was the case for the in situ hybridization data, which had a lower resolution, 200 micrometers, than the Allen Mouse Brain Coordinate Framework 29, 100 micrometers resolution. Region-level data, such as microarray gene expression data and RNA sequencing data, are typically encoding gene expression as count matrix 88, reporting the frequency of gene transcripts for samples. For dataset included in this study, these samples originate from brain regions. We manually map these brain regions to the corresponding brain regions of our reference ontology, Allen Brain Institute atlases, based on the region name and description in the dataset's reference publications. To ensure transparency and hence quality control, the detailed mappings are available in the resource's user interface. Browse database, then select a dataset to see details such as the dataset's mapping and in the supplemental material. The process of mapping region-level data to and retrieving it from a reference space is outlined exemplarily in Figure 7. Code for the mapping can be found in Supplementary Data 1. In this example, data are samples from the thalamus and hypothalamus, Figure 7a. Since the ontology maps to the corresponding voxels of the reference space, each voxel can be related to samples that originated from the voxel's brain region. The hierarchical nature of the ontology enables the querying of gene expression on multiple anatomical levels. For example, querying the average gene expression in the diencephalon, the parent region of thalamus and hypothalamus, will aggregate over all samples of the count matrix, Figure 7b, while a query on the thalamus or a subregion of the thalamus, e.g. dorsal thalamus, will result in an aggregation over the thalamic samples, Figure 7. C. We want to point out that thalamic samples do not necessarily represent dorsal thalamic samples, hence we make the sample's origin explicit in our resource's user interface, Fig. 5a. Sample region annotations. Figure 7. Mapping of exemplary RNA sequencing data to and retrieving from a common reference space. Samples of the thalamus, green, and hypothalamus, brown, of an exemplary RNA sequencing count matrix are mapped manually to a brain regions in an hierarchical ontology via literature research. Since mapping of the ontology to the reference space is known, samples can be mapped to individual voxels of the reference space, and hence to every anatomical level in the ontology. Be aggregating the average gene expression for all samples from a coarser anatomical level, diencephalon, than the original annotations, thalamus and hypothalamus. See aggregating the average gene expression for all samples from an equal or finer anatomical level, thalamus or dorsal thalamus, than the original annotations, thalamus, leads to the same results. Full-size image. We implemented 4 different variants of gene expression queries to cover different use cases, such as region specificity or enrichment. These queries were defined on a gene expression matrix of dataset D as 1, where each row represents a gene G as an element of G and each column a sample or a voxel in case of imaging data, S is an element of S, V subset of or equal to S represent all samples within the VOI. C subset of or equal to S samples of a certain cell type, and F subset of or equal to S A samples filtered by metadata other than cell types. Mean expression query. Computing the mean gene expression within the VOI for each gene G. 2. Region specificity query. To account for regional specificity, we compute for each gene G the mean gene expression within the VOI and normalize it to the mean expression of the rest of the brain. 3. Cell type specificity query. This query can be used to see how specific the expression of a certain cell type is. In this case, for each gene G, the mean gene expression within the VOI is computed for all samples of a certain cell type C subset of or equal to S and normalized by the expression over samples of all cell types within the VOI. 4. Enrichment query. This query can be used to see how specific the expression is for cell type C subset of or equal to S or different metadata F subset of or equal to S. In this case, the mean gene expression within the VOI is computed for all samples of the selected filter and cell type and normalized by the expression over all samples within the VOI. 5. BrainTrawler Lite. The basis of BrainTrawler Lite is the dataset coverage heat map, showing the distribution of samples/images across brain regions, columns, and datasets, rows, subdivided by metadata categories such as cell types, phenotypes, etc. Figure 5a. Regions can be dynamically set via a tree-like structure showing the brain ontology, Figure 5a left. Datasets can be subdivided by their metadata categories, e.g. split by cell types, Figure 5a, right, so that the rows do not only represent datasets, but also subsets thereof, e.g. a row for each cell type per dataset. Hovering over the individual tiles of the heat map reveals a summary of the data for the respective brain region and dataset, or subset, for example sample count and the original region annotation of the data. The original region annotation is especially relevant to identify the data's origin and hence the data's potential relevance for the user. In our first example, Figure 5b, one gene expression heat map is generated for each of the entered genes. Here, a gene expression heat map shows the averaged expression for all samples/images covered by each of the selected tiles in the dataset coverage heat map. This means that if the user selects a tile in the dataset coverage heat map of a certain brain region and a certain cell type of a certain dataset, each gene expression heat map will contain the same tile, showing the averaged expression of all the tiles covered samples/images, Figure 8a. To deal with gene lists with dozens of genes, we used a small multiples visualization 28, Figure 5b, right, so one can visually identify patterns while maintaining an overview. Clicking on individual gene heat maps will show a detailed view on the left-hand side, Figure 5. C left, displaying the exact expression values and row/column labels. The coloring is set by individual color scales per dataset, same color, but the range depends on the dataset's maximum value since, as already mentioned before, values are not directly comparable across datasets. Figure 8. Concept of how a selection in the dataset coverage heat map transfers to gene expression heat maps and the parallel coordinates system. Each tile represents a subset of the resource, i.e., samples/images of a certain brain region of a certain dataset, and of a certain metadata category. Each selected tile, orange, has a direct representation as tile in the gene expression heat map, A, and as axis in the parallel coordinates system, B. The values in A and B are the averaged expression values, e.g. CPM, TPM, etc. over all samples/images represented by the selected tile. Tiles without samples, missing data, are rendered gray in A or omitted for B. Full-size image. In the case of investigation on a genome level, Figure 5c, a parallel coordinate system is used analog to the dissection of connections shown in Figure 4. Here, each plot line represents a gene, indicating the averaged expression along axis for each selected tile, Figure 8b. Genes can be filtered by brushing along an axis, Figure 5c, to find genes with specific gene expression patterns. Spatial indexing. Real-time queries on the resource's data was achieved by spatial indexing, depending on the data type. Connectivity data. For real-time aggregation of connectivity data with billions of connections, we use the data structure introduced by Ganglberger et al. In 201927. The high query speed is reached by sorting rows and columns of a connectivity matrix by their location in space along a space filling curve 89, so that rows and columns that represent connections that are close together in the 3D reference space are also close together on the matrix axis. This makes reading local connectivity, e.g., the connectivity of a brain region, from the hard drive extremely efficient, since it benefits from read-ahead paging of the operating system to reach near-sequential reading speed 27. Imaging data. For computing the mean expression of a volume of interest, VOI, for example a brain region, we use spatial indexing on volumetric images similar to Scholl's 59. Here, the imaging data are not stored per image, but per voxel. For each voxel in the reference space, the data of all images at the voxel's position are stored together, i.e. on the physical hard drive. Furthermore, we order these per voxel data along a space filling curve 89, which allows data points in close proximity in the 3D reference to be stored in close proximity as well on the storage. The expression of the voxels of a VOI can then be read blockwise from the hard drive, which is more efficient than reading each image individually due to read-ahead paging of the operating system 59. Sample-based data. For sample-based data, such as RNA sequencing and microarray gene expression data, we used a similar approach as for imaging data. Here, for each sample in our resource, we use the samples mapping to the reference space, section mapping to a common reference space, to get the sample's location. Based on these locations, we ordered samples along a space-filling curve and stored them on the hard drive. This means that if a certain VOI is queried for gene expression, all relevant samples of all datasets are stored close together. As a consequence, they can be retrieved blockwise, benefiting from read-ahead paging of the operating system similar to the connectivity and imaging data approaches. We further optimize the queries by pre-aggregating samples with similar metadata, i.e. samples of the same dataset, cell type, age category, etc. This increases the query speed since the amount of data that need to be aggregated on the fly is reduced from thousands of individual samples to a tenth or even a hundredth of it, depending on the extent of the query. Data availability. BrainTrawler, including the BrainTACO resource, can be publicly accessed via BrainTrawler VRVista AT. All means to set up a custom BrainTrawler instance, including a Docker image, is available at Zenodo. https://doi.org/10.5281/zenodo.10400999. This does not include the third-party gene expression or connectivity data. All code to include these data, as well as custom datasets into BrainTrawler, is provided in Supplementary Data 1. See section Code Availability for details. Code availability. The software to operate BrainTrawler consists of a multitude of services, including spatial indices for gene expression and connectivity, a graph database, a web interface, and an API to handle individual parts 26. Hence, making the code open source does not necessarily guarantee that it can be easily used by others. However, we believe that providing a publicly accessible, free-to-use instance of BrainTrawler/BrainTACO, BrainTrawlered, VRVist, AT, as well as all means to operate a custom BrainTrawler instance, see section data availability, serves the scientific community best, as it allows researchers to leverage the data without the need for extensive technical knowledge. The code for the mapping and data generation of all gene expression or connectivity data of the BrainTACO resource is provided in Supplementary Data 1. In combination with the provided Docker image, see section data availability, it is possible to reproduce the public instance running at BrainTrawler, VRVist, AT, as well as to include one's own custom datasets. However, setting up a separate BrainTrawler instance may not be the most efficient approach for most researchers. VRVist, as a not-for-profit research center, is open to providing support in setting up a BrainTrawler instance in the scope of a research collaboration and is committed to further developing and improving the system to better serve the scientific community. We welcome initiatives for joint research and development projects and generally provide the software within these projects free of charge. References: Koch C. The Quest for Consciousness, A Neurobiological Approach, Volume 19, Roberts and Company Publishers, 2004. Kint, M. A Behavioral Neuroscience Perspective on the Etiology and Treatment of Anxiety Disorders. Behav. Res. Ther. 62, 24-36, 2014. Article PubMed Google Scholar. Griesner, J. et al. Central amygdala circuit dynamics underlying the benzodiazepine anxiolytic effect. Molecular Psychiatry. 26, 534-544, 2018. Young, L. J. and Wang, Z. The Neurobiology of Pair Bonding. Nat Neurosci. 7, 1048-1054, 2004. Article SAS PubMed Google Scholar. Allen Institute, https://www.alleninstitute.org 2019. Human Brain Project, https://www.humanbrainproject.eu 2019. Van Essen, D. C. et al. The WMIN Human Connectome Project, an overview. Neuroimage 80, 62-79, 2013. Article PubMed Google Scholar. Markello, R. D. et al. Neuromaps, Structural and Functional Interpretation of Brain Maps. Nat Methods 19, 1472-1479, 2022. Article SAS PubMed Central Google Scholar. Song, L. et al. Stab, A Spatiotemporal Cell Atlas of the Human Brain. Nucleic Acids Res 49, D1029, D1037, 2021. Article SAS PubMed Google Scholar. French, L. and Pavlidis, P. Relationships between gene expression and brain wiring in the adult rodent brain. Pilo S. Compot. File 7 E1001049 2011. Article SAS PubMed Central Google Scholar. G. S. Fakhri A. and Dang, H. Integrative Analysis of the Connectivity and Gene Expression Atlases in the Mouse Brain. Neuroimage 84, 245-253, 2014. Article PubMed Google Scholar. Ricciardi, J. and Altman, A. Correlated Gene Expression Supports Synchronous Activity in Brain Networks. Science 348, 11-14, 2015. Article Google Scholar. Rubinov, M. Itma, R. J. F. Watson, C. and Bullmore, E.T. Wiring Cost and Topological Participation of the Mouse Brain Connectome. Proc. Natal Academic, Sci. 112, 201420315, 2015. Article Google Scholar. Whitaker K. J. et al. Adolescence is associated with genomically patterned consolidation of the hubs of the human brain connectome. Proc. Natal Academic, Sci. 113, 9105-9110, 2016. Article SAS PubMed Central Google Scholar. French, L. Tan, P. C. and Pavlidis, P. Large-scale analysis of gene expression and connectivity in the rodent brain, insights through data integration. Front. Neuroinformatics 5, 12, 2011. Article Google Scholar. Fulcher, V. D. and Fernito, A. A transcriptional signature of hub connectivity in the mouse connectome. Proc. Natal Academic, Sci. 113, 1435-1440, 2016. Article SAS PubMed Central Google Scholar. Keel, J. M. Kali, A. and Kwan, K. Y. Brain Transcriptome Databases, A User's Guide. J. Nirasai. 38, 2399-2412, 2018. Article SAS PubMed Central Google Scholar. Cyber Explorer, https://atlases.ebrains.eu/viewer, 2019. Kaspers, S. et al. Studying variability in human brain aging in a population-based German cohort rationale and design of 1,000 brains. Frontiers in Aging Neuroscience, 6, 149, 2014. Horowitz, M. J. et al. An Anatomically Comprehensive Atlas of the Adult Human Brain Transcriptome. Nature 489, 391-399, 2012. Article SAS PubMed Central Google Scholar. Horowitz, M. et al. Canonical Genetic Signatures of the Adult Human Brain. Nat Nirasai. 1832-1844, 2015. Article SAS PubMed Central Google Scholar. Lawless et al. Exploration and visualization of gene expression with neuroanatomy in the adult mouse brain. BMC Bioinform. 9, 153, 2008. Article Google Scholar. Foam, D. et al. Exploration and visualization of connectivity in the adult mouse brain. Method 73, 90-97, 2015. Article SAS PubMed Google Scholar. O.S. W. et al. A mesoscale connectome of the mouse brain. Nature 508, 207-214, 2014. Article SAS PubMed Central Google Scholar. Line E. S. et al. Genome-wide Atlas of Gene Expression in the Adult Mouse Brain. Nature 445, 168-176, 2007. Article SAS PubMed Google Scholar. Ganglberger, F. et al. BrainTrawler, a visual analytics framework for iterative exploration of heterogeneous big brain data. Comput. Graph. 82, 304-320, 2019. Article Google Scholar. Ganglberger, F. Kaczynowska, J. Haubensak, W. and Bühler, K. A data structure for real-time aggregation queries of big brain networks. Neuroinformatics 18, 131-149, 2020. Article PubMed Google Scholar. Tuft, E. Envisioning Information. Graphics Press, 1990. Wang, Q. et al. The Allen Mouse Brain Common Coordinate Framework, a 3D Reference Atlas. Cell 181, 936-953, 2020. Article SAS PubMed Central Google Scholar. Fonov, V. et al. Unbiased Average Age-Appropriate Atlases for Pediatric Studies. Neuroimage 54, 313-327, 2011. Article PubMed Google Scholar. Gakio, et al. Cellular taxonomy of the mouse striatum as revealed by single-cell RNA-seq. Cell Republic 16, 1126 to 1137, 2016. Article SAS PubMed Central Google Scholar. Campbell, J. N. et al. A molecular census of arcuate hypothalamus and median eminence cell types. Nat Neurosci. 4, 2017. Google Scholar. Rossi M. A. et al. Obesity remodels activity and transcriptional state of a lateral hypothalamic brake on feeding. Science 364, 1271 to 1274, 2019. Article SAS PubMed Central Google Scholar. Bhattacharjee, A. et al. Cell type-specific transcriptional programs in mouse prefrontal cortex during adolescence and addiction. Nakkomun. 10, 4169, 2019. Guins N. W. et al. Integrated morphoelectric and transcriptomic classification of cortical GABergic cells. Cell 183, P935-953, E19, 2020. Yao, Z. et al. A taxonomy of transcriptomic cell types across the isocortex and hippocampal formation. SSRN Electron. J. 2020. Zeisel, A. et al. Molecular architecture of the mouse nervous system. Cell 174, 999 to 1014, 2018. Article SAS PubMed Central Google Scholar. Hodge, R. D. et al. Conserved cell types with divergent features in human versus mouse cortex. Nature 573, 61 to 68, 2019. Article SAS PubMed Central Google Scholar. Lee H. et al. Cell type-specific transcriptomics reveals that mutant huntingtin leads to mitochondrial RNA release and neuronal innate immune activation. Neuron 107, 891 to 908, 2020. Article SAS PubMed Central Google Scholar. Aguet, F. et al. The GTX Consortium Atlas of Genetic Regulatory Effects Across Human Tissues. Science 369, 1318 to 1330, 2020. Article SAS Google Scholar. Miller, J. A. et al. Transcriptional Landscape of the Prenatal Human Brain. Nature 508, 199 to 206, 2014. Article SAS PubMed Central Google Scholar. Nowakowski, T. J. et al. Spatiotemporal gene expression trajectories reveal developmental hierarchies of the human cortex. Science 358, 1318 to 1323, 2017. Article SAS PubMed Central Google Scholar. Darmanis, S. et al. A survey of human brain transcriptome diversity at the single-cell level. Proc. Natal Academic, Sci. USA 112, 7285 to 7290, 2015. Article SAS PubMed Central Google Scholar. Zhong, S. et al. A single-cell RNA-seq survey of the developmental landscape of the human prefrontal cortex. Nature 555, 524 to 528, 2018. Article SAS PubMed Google Scholar. Fan, X. et al. Spatial transcriptomic survey of human embryonic cerebral cortex by single-cell RNA-seq analysis. Cell Res 28, 730 to 745, 2018. Article SAS PubMed Central Google Scholar. Lee M. et al. Integrative functional genomic analysis of human brain development and neuropsychiatric risks. Science 362, 87615, 2018. Article SAS PubMed Central Google Scholar. La Mano, G. et al. Molecular diversity of midbrain development in mouse, human, and stem cells. Cell 167, 566 to 580, 2016. Article PubMed Central Google Scholar. Habib, N. et al. Massively parallel single-nucleus RNA-seq with DRONC-seq. Nat Methods 14, 955 to 958, 2017. Article SAS PubMed Central Google Scholar. Welch, J. D. et al. Single-cell multiomic integration compares and contrasts features of brain cell identity. Cell 177, 566 to 580, 2019. Article Google Scholar. Liu, S. J. et al. Single-cell analysis of long non-coding RNAs in the developing human neocortex. Genome Bio, 17, 1-17, 2016. Article Google Scholar. Onorati, M. et al. Zika virus disrupts phospho, TBK1 localization and mitosis in human neuroepithelial stem cells and radial glia. Cell Republic 16, 2576 to 2592, 2016. Article SAS PubMed Central Google Scholar. Lake B. et al. Integrative single-cell analysis of transcriptional and epigenetic states in the human adult brain. Nat Biotechnol. 36, 70 to 80, 2018. Article SAS PubMed Google Scholar. Zhao, S. Yi, Z, and Stanton are misuse of RPKM or TPM normalization when comparing across samples and sequencing protocols. RNA 26, 903 to 909, 2020. Article SAS PubMed Central Google Scholar. Evans, C. Harden, J. and Stubble, D. M. Selecting between sample RNA-seq normalization methods from the perspective of their assumptions. Brief. Bioinforma. 19, 776 to 792, 2018. Article SAS Google Scholar. Oss, T. et al. Anatomical variability, multimodal coordinate systems, and precision targeting in the marmoset brain. Neuroimage 250, 118965, 2022. Article PubMed Google Scholar. Larson, S. D. and Martone, M. E. Ontologies for Neuroscience. What are they and what are they good for? Front. Neurosci. 3. 60 to 67, 2009. Article PubMed Central Google Scholar. Ding, S. L. et al. Comprehensive Cellular Resolution Atlas of the Adult Human Brain. J. Comp. Neural. 524, 3127 to 3481, 2016. Article PubMed Central Google Scholar. Ding, S. L. et al. Allen Human Reference Atlas, 3D, 2020. RRID, SCR_017764, 2020. Scholes, F. Computational Methods Enabling Interactivity in Analysis and Exploration of Volumetric Images. PhD thesis, Institute of Computer Graphics and Algorithms, Vienna University of Technology, Favoritenstrasse 9-11/E193-02, A. 1040 Vienna, Austria, 2013. Hong, S. J. et al. Toward Neurosubtypes in Autism. File, Psychiatry 88, 111 to 128, 2020. Article PubMed Google Scholar. Fernito, A. Zaleski, A. and Breakspear, M. The Connectomics of Brain Disorders. Nat Reverend Neurosci. 16, 159 to 172, 2015. Article SAS PubMed Google Scholar. Fernito, A. and Bullmore, E.T. Reconciling Abnormalities of Brain Network Structure and Function in Schizophrenia. Kerr, Open, Neurobiol. 30, 44 to 50, 2015. Article SAS PubMed Google Scholar. Ripk, S. et al. Biological insights from 108 schizophrenia-associated genetic loci. Nature 511, 421 to 427, 2014. Article SAS PubMed Central Google Scholar. Just S. and Geschwind, D. Disentangling the heterogeneity of autism spectrum disorder through genetic findings. Nat Reverend Neural, 10. 74 to 81, 2014. Article PubMed Central Google Scholar. Zerby, V. et al. Brain mapping across 16 autism mouse models reveals a spectrum of functional connectivity subtypes. Molecular Psychiatry, 26, 7610 to 7620, 2021. Schreederin, D. Levitin, D. J. and Menon, V. A critical role for the right frontoinsular cortex in switching between central executive and default mode networks. Proc. Nal Academic, Sci. 105, 12,569 to 12,574, 2008. Article SAS PubMed Central Google Scholar. Menon, V. et al. Optogenetic stimulation of anterior insular cortex neurons in male rats reveals causal mechanisms underlying suppression of the default mode network by the salience network. Nakkomun, 14, 866, 2023. Article SAS PubMed Central Google Scholar. Craig, A. How do you feel now? The anterior insula and human awareness. Nat Reverend Neurosci, 10, 59 to 70, 2009. Article SAS PubMed Google Scholar. Gagala, N. The insular cortex. Kerr, File, 27 R580, R586, 2017. Article SAS PubMed Google Scholar. Sheffield, J. M. Rogers, B. P. Blackford, J. U. Heckers, S. and Woodward, N. D. Insula functional connectivity in schizophrenia. Schizophrenia Res 220, 69 to 77, 2020. Article Google Scholar. Nomi, J. S. Molnar-Jacotz, I. and Udine, L. Q. Insular function in autism. Update and future directions in neuroimaging and interventions. Prague. Neuropsychopharmacol. File. Psychiatry 89, 412 to 426, 2019. Article Google Scholar. Uter. S. Bull, S. Eipert, F. and Butchell, C. Functional dissociation of stimulus intensity encoding and predictive coding of pain in the insula. eLife 6, E24770, 2017. Article PubMed Central Google Scholar. Bauernfeind, A. L. et al. A volumetric comparison of the insular cortex and its subregions in primates. J. Hum, EVOL. 64, 263 to 279, 2013. Article PubMed Central Google Scholar. Ochoa, D. et al. The Next Generation Open Targets Platform. Reimagined, redesigned, rebuilt. Nucleic acids Res 51, D1353, D1359, 2022. Article PubMed Central Google Scholar. Stark, Z. Bruno, D. L. Mountford, H. Lockhart, P. J. and Amore, D. J. De novo 325 kb microdeletion in chromosome band 10q 25.3, including atenyl 1 in a boy with cognitive impairment, autism, and dysmorphic features. Your J. Med. Janet, 53, 337 to 339, 2010. Article PubMed Google Scholar. Gonzalez Burgos, G. and Lewis, D. A. GABA neurons and the mechanisms of network oscillations. Implications for understanding cortical dysfunction in schizophrenia. Schizophrenia Bull. 34, 944 to 961, 2008. Article Google Scholar. Keelik, F. Isaac, U. USTA, A. and Demertas, A. Serum tumor necrosis factor-like weak inducer of apoptosis levels are elevated in schizophrenia. Revista Brasileira Psiquiatria 43, 2020. Schmidt, F. M. et al. Ligands and receptors of the TNF superfamily are decreased in major depression and during early antidepressant therapy. J. Psychiatr. Res 119, 116 to 121, 2019. Article PubMed Google Scholar. Karadeg, H. et al. Serum TNF-related weak inducer of apoptosis, tweak, TNF-related apoptosis-inducing ligand, trail, levels in patients with bipolar depression, major depression, and a healthy control group. Psychiatr. Danubina 33, 314 to 319, 2021. Article SAS Google Scholar. How, why, et al. Integrated analysis of multimodal single-cell data. Cell 184, 3573 to 3587, 2021. Article SAS PubMed Central Google Scholar. Carlson, M. org.hs.eg.db, Genome-Wide Annotation for Human, 2020. Arnakovichet, A. Fulcher, B. D. and Fernito, A. A Practical Guide to Linking Brain-Wide Gene Expression and Neuroimaging Data. Neuroimage 189, 353 to 367, 2019. Article PubMed Google Scholar. Zaldivar, A. and Kritchmer, J. L. Allen Brain Atlas-Driven Visualizations, a web-based gene expression energy visualization tool. Front. Neuroinformatics 8, 51, 2014. Article Google Scholar. Ganglberger, F. et al. Predicting functional neuroanatomical maps from fusing brain networks with genetic information. Neuroimage 170, 113 to 120, 2018. Article PubMed Google Scholar. Skok, A. et al. Human brain structural connectivity matrices, ready for modeling. Sci. Data 9, 486, 2022. Article PubMed Central Google Scholar. Duve, L. Dune, K. V. Dusseldorp, E. and Mechelin, I. V. Quint, a tool to detect qualitative treatment, subgroup interactions in randomized controlled trials. Sciother, Res 26, 612 to 622, 2016. Article PubMed Google Scholar. Avance B. et al. A reproducible evaluation of ANT's similarity metric performance in brain image registration. Neuroimage 54, 2033 to 2044, 2011. Article PubMed Google Scholar. Malhotra, A. Das, S. and Rai, S. N. Analysis of single-cell RNA sequencing data, a step-by-step guide. Biomedinformatics 2, 43 to 61, 2021. Article Google Scholar. Hilbert, D. Über die Städtische Bildung einer Line auf ein Flaschenstock. Mathematisch and 38, 459 to 460, 1891. Article Google Scholar. Download references. Acknowledgements. VRVIS is funded by BMK, BMW, Styria, SFG, Tyrol, and Vienna Business Agency in the scope of COMET, Competence Centers for Excellent Technologies, 879730, which is managed by FFG. Research on spatial indexing was funded by the Austrian Science Fund, FWFI 4836. B. Wulf Haubensak was supported by the Research Institute of Molecular Pathology, IMP, Boehringer Ingelheim, the Austrian Research Promotion Agency, FFG, and a grant from the European Community's 7th Framework Program, FP/2007-2013/ERC Grant Agreement Number 311701. The extensions of BrainTrawler with the integration of transcriptomic data were funded by Boehringer Ingelheim, where we want to specifically thank Till and Lauer for his writing support, and Moritz von Heimendel, Roberto Arbin, Frank Gillardin, Sergio Picard Armada, Grigiorio Alanis Labato, and Yassine Kamaz for the selection and assistance with the datasets. Florian Ganglberger performed his work for the paper as postdoc employed at Viarvis, but finalized and submitted the paper as principal scientist at Boehringer Ingelheim, RCV GmbH, and Co. KG. Furthermore, we want to thank Piotr Radkowsk and Adam Philipp, Artigen, for pre-processing, Nicholas Swoboda, Viarvis, for web development support, as well as Sofia Yolonska and Johannes Novotny, Viarvis, for proofreading and support during the review process. Author information. These authors contributed equally: Dominic Kargl, Markus Töpfer. Authors and affiliations. Biomedical Image Informatics, Viarvis Research Center, Vienna, Austria. Florian Ganglberger, Markus Töpfer and Katja Bühler. Global Computational Biology and Digital Sciences, Boehringer Ingelheim Pharma, Biberach and der Riss, Germany. Florian Ganglberger, Julien Hernandez-Lallement, Nathan Lawless, and Francesc Fernandez-Albert. Department of Neuronal Cell Biology, Vienna Medical University, Vienna, Austria. Dominic Kargl and Wulf Haubensak. Research Institute of Molecular Pathology, IMP, Vienna Biocenter, VBC, Vienna, Austria. Wulf Haubensak. Contributions. FG was the main author behind the development of BrainTrawler, including its conception and the mapping and processing of the datasets. FG also played a significant role in the creation of BrainTrawler Lite, a task shared with MT. Both FG and MT were involved in the coding and development of BrainTrawler, MT for BrainTrawler Lite. DK and WH created the case study, contributing to its discussion and interpretation. Their insights were crucial to the understanding and analysis of the study. J. Chell was responsible for the anatomical mapping of the datasets to the reference space. In addition to this, J. Chell also provided writing support, ensuring the clarity and coherence of the paper. FG, NL, and FA conceived BrainTACO as a resource. N.L and F.F.A. S input was also valuable in the development of BrainTrawler Lite. KP played a supervisory role throughout the project, providing guidance and oversight. Corresponding author. Correspondence to Katja Bühler. Ethics declarations. Competing interests. The authors declare no competing interests. Peer review. Peer review information. Communications Biology thanks the anonymous reviewers for their contribution to the peer review of this work. Primary handling editors, Saba Besharati and Mengtan Xing. A peer review file is available. Additional information. Publishers note Springer Nature remains neutral with regard to jurisdictional claims in published maps and institutional affiliations. Supplementary information. Peer review file. Supplementary information. Description of additional supplementary files. Supplementary data. NR reporting summary. Rights and permissions. Open access. This article is licensed under a Creative Commons Attribution 4.0 International License, which permits use, sharing, adaptation, distribution, and reproduction in any medium or format. As long as you give appropriate credit to the original authors and the source, provide a link to the Creative Commons license and indicate if changes were made. The images or other third-party material in this article are included in the article's Creative Commons license, unless indicated otherwise in a credit line to the material. If material is not included in the article's Creative Commons license and your intended use is not permitted by statutory regulation or exceeds the permitted use, you will need to obtain permission directly from the copyright holder. To view a copy of this license, visit http://creativecommons.org/licenses/by/4.0/ Reprints and permissions. About this article. Cite this article. Ganglberger, F. Kargl, D. Töpfer, M. et al. BrainTACO, an explorable multi-scale multimodal brain transcriptomic and connectivity data resource. Commune Vial 7, 730, 2024. https://doi.org/10.1038/S42003-024-06355.